One of the more controversial topics in biotechnology is cloning. And cloning is making an exact copy, genetically identical copy, of a cell, a gene, or an organism. And I think most people, when they think of cloning, immediately think of cloning an organism like Dolly. But there's actually a couple different kinds of cloning, and some are certainly more controversial and less controversial. So first we have cloning a gene, where you copy a single gene. We have cloning a cell, where you make a genetically identical copy of a single cell. And then we have cloning entire organisms, where you create a new individual that has the same genotype. Cloning a gene is a technology that's used in biotechnology all the time. We just talked about recombinant DNA, and we're going to make a copy of a gene, put that into a plasmid, make many copies of the plasmid, and have those invade new bacterial cells or other target cells. And again, this is a normal technology that's used all the time in scientific research and production of medicines. And it's not particularly controversial. Cloning a cell, we're going to learn pretty soon the process of mitosis. Mitosis is when one cell makes a genetically identical copy of another one, and our body does this all the time. So you have cell cloning happening naturally inside of you. There can also be therapeutic cloning, and this is done by scientists, not the natural cloning of a cell. And this is used primarily to keep stem cell lines going for research. If you have a petri dish full of cells and those cells are important for your research, you need to continue to have that line. You want to continue to clone those. Let's say you've developed a particular kind of cell that you're using to test breast cancer drugs on. You want all of those cells to be exactly genetically identical so that when you're running your study, you know that any difference you see in the survival of those cells, it's due entirely to the medications that you are subjecting those cells to, not genetic differences among different cells. The controversial kind of cloning is cloning an organism. When you clone an organism, you take the nucleus of a cell from an adult full-grown organism and you put it into the nucleus of an egg cell, and you let that egg develop with that complete nucleus, and you will end up with offspring that are genetically identical to that initial nucleus. Dolly the sheep was probably one of the most famous early examples of successful cloning. We have not successfully cloned a human being, and right now there's not a lot of research going into trying to do that. The main groups that are really truly interested in cloning organisms are in agriculture, where you're trying to find cows that produce lots of milk, or pigs that grow really quickly and produce really good tasting meat. And could we clone those individuals as a way of ensuring that we continue to get those products? Bill Nye does a great job of explaining the different sides of issues, and so I'm going to have you watch another Bill Nye episode, this one about cloning, and it's got some interesting background and theoretical questions. You can pause now and watch the video, or continue to fill out your lecture study guide and then come back to this video. Bioinformatics. This is using computers and automation to speed up the analysis of large volumes of biological data. We're at a really interesting point in science where, for the first time, we have more data than we have time and people to analyze. Historically, if scientists had a question, they went out and collected data, but data was often the limiting factor. We're now able to collect large, huge volumes of data. We have the Human Genome Project. It now takes about 3 weeks to do the genome of a new organism, and we have so much information and not the people or the time to comb through all of that data. And so there's this growing field of bioinformatics that brings together computer analysis and automation to help us deal with all of that data. Bioinformatics uses something called high throughput technology, which includes really fast computers, robots, and automated sequencers. So we take a lot of the day-to-day tasks away from people and leave the analysis piece to the human brain, which is good at that part. So you're going to watch two videos about this. Here we have a math professor talking about some of the work his students are doing to help analyze biological data. And here we have two researchers talking about how they can bring together this technology to move forward our understanding of human health and disease. We actually have a biotechnology program here at American River College, and you can get a certificate in biotechnology. It gives you more detailed background in everything that we've talked about in this lecture, and it gives you some lab experience in some of the techniques that you need to actually do some of this work. And believe it or not, Northern California is a hub for biotechnology, especially associated with UC Davis. There's a lot of plant-related biotechnology, and Genentech is a huge company. Monsanto is another company that does a lot of biotechnology kind of research. And then down in the Bay Area, there are many biotechnology-related firms. So, if it sounds interesting to you, I would suggest looking into their classes, and there's even one specifically about bioinformatics and understanding how computers can be used to help analyze this kind of data.